Morning, Dr. Taylor.
Good morning. Let's see.
How's everything going?
I'm just hearing you now. My earphone wasn't connected. How we doing?
Doing pretty good. Pretty excited to present today. So, yeah.
How many presentations will there be?
I believe 3 or 4 in total.
Uh-huh.
Yeah. All of them I think are about 10 minutes each, and then there's like 5 or 10 minutes of Q&A or discussion, maybe a little shorter than that. But yeah, I would say like 3 or 4 presentations.
Okay.
Yeah.
And how many are on your team?
So we basically have a total of 12 people on our team, but today I think only not everyone is going to be there. There's some that have other commitments today, so I think it's going to be around, I didn't count. I want to say a little less than 10. So I want to say at least 9 people on our team are going to be showing up today to present.
So are you going into your senior year?
Actually, my junior.
Oh, your junior year. Okay.
Do you teach any classes by any chance?
Yeah. I'm this semester I will be teaching a course I've taught for, I don't know, 25 years. Environmental microbiology.
Oh, that's awesome.
Yeah. So it has very, I mean, it focuses on microbes in the environment as opposed to clinical and disease and all of that. Yeah. But we do touch on public health and epidemiology as well.
Wow, that's amazing.
Yeah.
It sounds really interesting.
And it has a lab. It has a 3-hour lab. And we get a lot of bio majors because it's an upper division lab course. It's 301. So I would say 90% of our students are pre-health and bio majors as opposed to environmental types and marine sciences.
Wow, that's amazing. What's the class number for that? I'm actually kind of interested.
We're already over-enrolled, oversubscribed.
Yeah, I hope in the future.
Oh yeah, so next year, MAR 301.
MAR 301. Okay, that sounds
environmental microbiology. Yeah.
Gotcha. That's really nice. And yeah, environmental microbiology is very relevant for today's presentation. So that's perfect. Hey, Lamisa.
Hello.
Good morning.
Morning.
For those of us in America.
Oh yeah, and this is Lamisa, by the way, our outreach leader.
Hi, Lamisa.
Nice to meet you, Dr. Taylor.
Nice to meet you too. So this course that I teach, I've been using the same textbook the entire time, and it gets updated every 3 years, I think. And so this last spring, it got updated again, and I ordered it for students. And when I went online yesterday, I discovered that it's only available digitally as a rental. Students can, at least I have not found a way to order the hard copy if students actually want to have a physical book. And this is a first for me. I've never had a publisher do this. And I don't know how it's going to go over. I'm trying to have both formats available and I'm not having any success, but I just discovered it yesterday. So hopefully I can get it resolved on Monday.
Gotcha.
It's making me a little nervous.
Right. I mean, generally, from what I know, like a lot of companies, like a lot of textbook companies, they have a hard copy available as well as a digital copy, but they charge more for the hard copy.
A lot more. A lot. Yeah, they're expensive. I know.
Yeah. So, obviously, like I can't buy the hard copy, so like I have to adapt to just reading a digital textbook. So that was not fun, but hey, I mean, I had to.
Yeah. There's another member of the team that's joined us, Ashley. Hi.
Hello, sir. How are you? We're so glad that you're here today.
Thank you.
Thank you for joining us.
You're welcome. You're chained to the lab? Is that a
Yes, sir.
virtual background, or are you really in the lab?
I'm really in the lab.
Okay.
We just joined the lab like 5 days earlier, so we're all very excited to just stay here for as long as we can.
Okay.
Yeah.
Was there an iGEM last year? Was it canceled because of COVID?
The jamboree was canceled. It was virtual, but we did have Stony Brook did have an iGEM team last year. Yeah.
Uh-huh. Okay.
I
And
think
were you,
oh, sorry, go.
I was going to say, were you all on it last year?
We're all new except for one member, Abhishek. I don't know if you know him, but
No, I don't.
yeah, but the rest of us are all new to
Okay.
iGEM.
Yeah, so the program I think has been going on 8 or 9 years strong at this point. So yeah.
How many different countries participate?
I think I don't know. I know there's over 300 something teams.
Uh,
I don't exactly remember the countries, but it's from all over the world. So
Yeah, I
Yeah.
looked at the website a little bit, and I saw that there were lots of dots on the map.
Mhm.
Yeah.
It's a very cool experience.
Yeah. So other than the jamboree, are there periodic get-togethers where everybody's communicating and?
It's more like a virtual get together. I know there are sometimes meetups. Like I know there's some in Boston and Harvard's doing one this year.
Uh-huh.
In terms of like North America, but Jamboree is usually the point where like everyone meets together in person. But this year it's also going to be virtual.
Interesting. Hello.
Good morning, everyone, or afternoon, or
Good morning.
Good
morning.
day. Have a good day.
day.
Good day.
That's the right
Hello, am I audible?
Yes.
Hello, I hope you all are doing well. I am Vidisha
And I'm Arsh.
we both are from team IISER-Pune-India and we'll be your host for the day. First of all, thank you for being here with us today. It is really nice to see everyone here. So let's get started. Firstly, we'd like to welcome all of you to the day one of our two-day Cyanobacteria Symposium. Today's event involves project presentation by Cyanobacteria team, followed by critical feedback from our panel of experts.
Working with cyanobacteria has proven to be hard, mostly because of lack of proper guidance. After an exhaustive literature review, all iGEM teams presenting today have come up with very innovative projects. However, there are still a lot of concerns to be addressed and many holes to fill. Hence, teams IISER-Pune-India, Stony Brook, Hong Kong HKU, and Toulouse INSA-UPS have organized a project presentation session with experts from all over the world for teams working on cyanobacteria.
So for today's event, every team will get 10 minutes to present their projects, followed by a 10-minute expert comment session. After which the judges will have 5 minutes to fill the feedback forms for the presentation. We would request everyone present here to respect everyone's time.
And finally, we are thrilled to announce that we have contacted the best in their fields. We have Dr. Daniel Ducat, Professor Himadri Pakrasi, and Professor Gordon T. Taylor to judge these presentations. We'd like to welcome them to today's symposium. We would also like to mention that Dr. Tiago Selão will be joining us today to witness some of the presentations. We will also be seeing him tomorrow for his talk on a newly discovered cyanobacteria strain. I'd once again like to welcome all our experts, and without wasting any more time, let's start with our first presentation.
So our first presentation is brought to you by Team Stony Brook, who will be speaking on bioremediation of microcystin in lake drinking and recreational water. I hereby request Team Stony Brook to come and present their project. You guys will be having 10 minutes for the presentation, followed by 10 minutes for expert advice.
Right, thank you so much. Can I share my screen? Or
Yeah, sure.
oops, clicked on the wrong thing. Okay, there we go. All right, hi everyone. So as we've been introduced, we're the 2021 Stony Brook iGEM team, and our project this year is on biodegradation of microcystin. So my name is Lamisa. I'm the outreach leader for my team, and the rest of my team members will introduce themselves as we're going through with the slides. So to understand microcystin, we first need to understand cyanobacterial harmful algal blooms, which are unwanted overgrowths of algae. And these are caused by favorable conditions such as climate change, so increasing temperature, and then excess nutrients such as nitrogen and phosphorus, which are often produced in excess due to fertilizers. So these disrupt drinking and recreational water access because they release toxins, which cause severe illnesses and death in humans, and also are a big threat to aquatic and avian wildlife. So microcystin is one such of those toxins that are the most common, and specifically MCLR is a type of microcystin, which is a hepatotoxin and carcinogen, and it is the most toxic, which is what we're focusing on. So there are already past methods of degradation of microcystin, such as ozonation, chlorination, and granular activated carbon. However, these are either very expensive or they produce toxic and corrosive byproducts, which then need to be filtered more to produce drinking water. So there are also past teams who have worked on a similar project, and we're hoping to improve on those projects as well.
MAI, which is a microcystinase that catalyze the first reaction of the biodegradation of microcystin, and it's a transmembrane protein and able to linearize the microcystin and therefore decrease the toxin like 2,000-fold. MLRB and C can further degrade the linearized microcystin to a shorter peptide. So like we are trying to like overexpressing the mRNA in E. coli and that could be used in a bioreactor to remove the microcystin, and it's more environment friendly and more cheaper. Yeah. Next slide.
All right, so these are basically our proposed solutions. So we're creating 3 constructs with our chassis as E. coli, since it's portable and has a quick generation time. It's also much easier to obtain than some other bacteria. So we have 3 constructs. The first 2 is for overexpression of MLRA. One will have MLRA anchored to the outer membrane. Construct 2 will have over-secrete MLRA in the periplasm, and construct 3 is for detection. So we'll go into more detail in the next couple of slides.
So since, like Jahan said, we want to overexpress the MLRA enzyme to linearize microcystin, one of our proposed methods is basically having our enzyme, our MLRA, anchored to the outer membrane of E. coli, or specifically BL21 strain. So basically we use a protein called PGSA, which is an anchoring motif, which basically attaches the MLRA onto the outer membrane. And so this surface display of enzymes can be beneficial because the substrate doesn't have to diffuse through the membrane to go inside E. coli. It could just, if microcystin is within the perimeter of E. coli, if it's near the surface of the bacteria, it could basically degrade. So how we plan to do this is basically by creating a fusion protein between PGSA and our enzyme MLRA. And this fusion protein will express the enzyme onto the outer membrane of E. coli. And theoretically, this should lead to faster degradation. Next slide. And this right here is our plasmid that we tried to work on.
Okay, hi everyone. My name is Nicole. I'm from Wet Lab, and I'm going to be describing our second strain. So when we spoke to professors, they informed us that it would be difficult to predict what method would be the best to have the best results. We couldn't predict that, so we decided to kind of have 2 separate approaches to microcystin degradation, just to kind of be on the safer side. So our second construct is going to have MLRA secreted to the periplasm. The reason why is that MCLR is a really small protein. It's just 7 amino acids, and it freely diffuses in and out of the periplasm. So we thought that if we could kind of saturate the periplasmic space with MLRA, microcystin could basically diffuse in and out and be broken down in that space, and potentially, without relying on like an anchoring motif or anything, we could have more MLRA just free floating in that space. The way that we're doing this is by adding a strong IPTG inducible promoter upstream of the TAP machinery, which are proteins that translocate fully folded proteins from the cytoplasm to the periplasm. It's really promising because we know that MLRA is fully folded and functional in the cytoplasm. It's just stuck in the cytoplasm. Adding the strong inducible promoter will let us have like more TAP machinery, so we can secrete more than E. coli naturally can, allowing for a higher export system. And we're using the methodology of Dr. Browning, who created this TAT express strain. We're actually using his procedure exactly, and they found that they had 5-fold increase in secretion. So we expect to have a lot of MLRA at the end of this. And then after that, the TAT express strain that we create would be transformed to express MLRA with a TORA signal peptide. That would be like a secondary transformation. And then that signal peptide would direct MLRA to this like secretion machinery. and this is just the backbone for it. And originally we didn't have access to the plasmid or the PDOC plasmid, and we actually had to ask a professor to directly ship it to us. So no longer was it a costly issue for us. Next slide.
So our third construct revolves around detection, and here we're using a novel 2-hybrid bacterial assay combined with proteins used from a commonly used assay for microcystin detection, the PP1 assay. So here we're looking at CAM production, which is stimulated by interactions between two adenylyl cyclase domains, T25 and T18. These two proteins are attached to our proteins of interest, which are PP1 and GSH, two proteins that can interact with microcystin. Microcystin is able to inhibit PP1, and GSH or glutathione actually detoxifies microcystin. So within the presence of microcystin, PP1 and GSH are able to connect to it through the ADDA site and MDHA site. And this reconstitutes adenylyl cyclase, which can then drive CAMP synthesis, activating our genes of interest, which we use a reporter gene, GFP. And this methodology was kind of inspired by the iGEM Tianjin 2009 team, which showed that the proximity of PP1 and GSH was able to bring the two domains close enough to stimulate CAM production. Next slide. And here we have our construct. So we have the components PP1 and GSH, as well as T18 and T25 linked together.
Hi everyone, my name is Assad and I'm a part of the dry lab and modeling team. So essentially the dry lab and modeling team needs to take the results that we get from wet lab. As Hira stated, the third construct has to deal with detection. Therefore, we're gonna detect how much microcystin is left and how well the first two constructs are able to degrade microcystin. Now we need to compare this to a theoretical standpoint and truly verify if our plasmids have worked. So the dry lab is responsible for this, and the dry lab team will validate the practicality of this mechanism using differential equations to actually model how much microcystin degrades theoretically at a certain point. This gives us a lot of insight on microcystin degradation as a whole, the activity of microdegradation over time, essentially the overall kinetics of the reaction. Next slide, please.
All right, so the method for using a packed bed bioreactor helps to encapsulate and immobilize E. coli cells into alginate beads, and this allows for water to flow through the beads, thus degrading the MCLR. Using a contaminated system minimizes the risk of releasing the genetically modified bacteria into the external environment. Finally, there's a lot of potential for this product to be used in emergency purification of lake water, as well as in treatment plants.
All right.
And
So we're done. So any questions, any feedback, anything is appreciated. We'll stop sharing now.
Thank you, Team Stony Brook, for your presentation. We will now move on to the expert comment session. I request one of our experts to start off by giving their inputs on the presentation.
All right, I will start. Very nice presentation. I think that there are tremendous possibilities here. A general question, how do you compare your approach with the chemicals that have been used in the past? I'm in particular thinking about hydrogen peroxide that was used earlier last decade by a Dutch group that showed that took care of the algal bloom quite well. If your experiments are successful, where would that take you in comparison to those chemical approaches?
So I'll start. So basically, our project specifically aims to degrade the toxin microcystin, more specifically, which is more of a product formed by harmful algal blooms. So that's kind of, so basically we're kind of, our team wants to kind of sort of focus and work on a specific issue when it comes to biodegradation. And also how it compares to other teams, how it compares to like past projects is, so we kind of briefly went over this in one of our slides, that that one of the past papers, they expressed the enzyme MLRA in the cytoplasm of E. coli heterologously. So basically what we want to do is kind of improve upon their methods and sort of express it on the outer membrane or have it expressed in the periplasm, which should allow, in theory, for much faster degradation of microcystin, which could be used in a biofilter, basically. So basically, we just hope to use this to clear toxins from water, because this toxin is carcinogenic and has a lot of harmful health effects. So that's why we want to target it.
Very nice presentation, guys. One of the early slides that you showed was an aerial view of Lake Erie in the middle of a microcystis bloom, which was pretty horrendous. The strategy that you folks are coming up with would be targeted for local water use rather than environmental cleanup. What do you know about the environmental abundance of microcystinase? Are there many bacteria out there that actually express this enzyme, and might we look to enhance their activity out in nature?
I can answer this. There are actually like a lot of bacteria that express MLRA, and actually different combinations of MLR A, B, C, and D. One paper we looked at had like a whole page list of just like at least 20 different species and different combinations of genes that they express from this cassette. And they're all like slightly different, but overall it's the same type of protein that's being expressed. And in nature, some combinations of MLR A, B, C, D, and E basically will naturally break down microcystin. And some studies did find that in some lakes there are kind of combinations of like MCLR producing and MLR ABCD cassette expressing bacteria that kind of cancel out or end up cleaning up the water. The issue with that is that kind of in nature currently, it takes certain combinations of bacteria with some, but not all of these enzymes. So it's a little bit complicated to just kind of, for us, for like industrial applications, just like co-culture and guess at which combinations of bacteria will work. The paper that we read said that it is like a little bit finicky, like what species will work together in terms of enhancing like enhancing MLRA expression in nature. That would be really cool. Unfortunately, for like within the scope of iGEM, we're not allowed to consider any sort of release related projects, and everything has to be enclosed. But I mean, like hypothetically, I think that could work, as there are like small scale examples of that already happening. If we could kind of amp that up in nature, that would likely help a lot, but it's outside the scope of what's allowed within iGEM.
And to add on to what you said with the cassette, initially we did look at like MLRA B, C, and so on, but the further down you go, like down the cassette, the less research there is on that, since it is something that's pretty recent in the field. Like we found a paper on MLRA B and C that came out this year, and before that there wasn't much research on that, which is why we decided to just stick to MLRA for now, and then in the future hopefully we can advance onto more of the cassette.
Yeah, thank you.
Great presentation. It was extremely clear. Thank you. I was curious if when you're expressing these different forms of MLRA and targeting it to different compartments in E. coli, if you had a approach to try to detect where it is and is it really getting to the right subcellular localization?
Yeah, I can answer that. So basically we have a his tag on our MLRA. So basically with that, we are able to detect and make sure that, well, number one, if our MLRA is being expressed at all, but also it would let us know if it's also being expressed in the correct place. So we got our MLRA gene from the iGEM registry. Because past teams have worked with MLRA, obviously we're just trying to improve upon the methods. And those past teams have used his tags on their genome to detect MLRA. So basically, we're just going to be kind of doing the same thing. I guess there's no point in reinventing the wheel. So yeah.
So you would try to sort of fractionate out the different compartments and use, okay.
Yeah.
Nodding. And then this is a trickier question, but this information might not exist out there, but is there any information on how stable MLRA is in different pH regimes, for example, right? If you have it on the outer membrane, you no longer have this sort of controlled cytosolic environment. And have you thought about that at all?
Yeah, so basically, the thing is that MLRA in its native species, Sphingomonas, is an integral membrane protein. So basically, it is kind of like an outer membrane protein in its native species. So it's pretty stable in those conditions. But when it was expressed in 2012 in E. coli, it was actually expressed in the cytoplasm. So basically from that information, we were able to kind of infer that it should be stable in pretty much most environments. Again, when it comes to research on the cassette of the MLR proteins, there's still very limited research. Just recently, we know the crystalline structure of MLRA. Even though more research could be done, we do know that it's a fairly stable protein.
To add on to what Nabil said, there's actually a lot of questions about like how exactly MLRA works. And yes, like natively it's an integral membrane protein, so it should be stable in like a hydrophobic environment. And it probably shouldn't work the best in like an aqueous environment like the cytosol. But when it was tested in E. coli and the cytosol, researchers found that it worked, and there isn't really an explanation for why this protein assumes a stable conformation that's functional in both locations. And there really wasn't like any explanation. They were just kind of like, well, this is how it is. So going off that, we're basically kind of taking a stab in the dark and saying, oh, well, hopefully if it's kind of versatile and that it can work in a hydrophobic and hydrophilic environment, hopefully it'll also remain stable, exposed to like more varying conditions in the external environment, since we're not really sure why it's been so versatile in the past.
Okay, cool, thanks.
So let me ask a follow-up on that, Nicole. All of you are talking about if it works. How do you know that it worked? How do you know that the protein that you're expressing is functional?
We were gonna, after we have our constructs all set and done, we were gonna expose them to microcystin and then use a PP1 assay, basically like to see like the amount of microcystin to begin with and then follow its degradation. And that's something that past teams have done. So nothing new here. We're kind of taking inspiration from them there.
Good.
So yeah, just to kind of add on to that, our PP1 assay is just basically using spectrophotometry to essentially detect our microcystin degradation. So that's how we're going to be testing the success or lack thereof of our constructs.
Great. So we've run out of time for the comment section, and we'll now be moving on to filling the judging form. So I request all the judges to start filling the judging form. We have 5 minutes for that. In the meantime, I would request Team Hong Kong HKU to get ready for their presentation. You'll be presenting next. Are the judges done filling the form?
Almost.
Yeah, sure. Great. So I think it's time to start off with the next team. So coming up next is team Hong Kong HKU, who will be talking about a PET degrading bioreactor containing a symbiotic co-culture of E. coli and cyanobacteria. So you can go ahead and present your screen. You'll get 10 minutes for your presentation.
Okay, thank you, Arsh. Let me just share my screen first. Okay, so can you all see my screen?
Yes.
Yep.
Okay, good. Let's start presenting.
So good morning, good afternoon, and good evening to our guest judges, iGEM teams, and participants. I'm Isabella, and I'm a member of the iGEM team Evolve from the University of Hong Kong. This year, our project name is E. coli, and we'll be using cyanobacteria and E. coli to address the plastic problem. But before we go into the details, let me introduce and illustrate the problem our team is planning to address. So the global plastic problem is worsening. I'm sure you're all no stranger to this fact, and that there is also no quick solution for it. Even if there are ways to degrade plastic, they tend to be ineffective and cannot be implemented on a larger scale. In 2019 alone, 17 million tons of PET were used for packaging and textile. And in textile, PET is commonly known as polyester. And for comparison, that's as heavy as 17.5 million elephants combined. On top of that, almost 80% of it ends up in the landfill, which are harmful to life and the environment. But plastic is obviously a useful material, and we can't change consumer behavior. Therefore, we are proposing an alternative, a synbio-based solution to this problem. Our team aims to produce a solar-driven, self-sustaining bioreactor to digest polyethylene terephthalate, or PET for short, utilizing the potential symbiotic relationship between the bacteria E. coli and Cyanobacteria Sinococcus elongatus. We hope to synthetically develop a co-culture system between them where we can repurpose textile, packaging, and PET waste. So basically what we're trying to do here is to alter the life cycle of PET instead of dumping it straight into the landfill after one use. And so we want the PET to be degraded into monomers and processed into new bioplastics. So let me pass the time to William, who will explain the design behind the project.
So hi everyone, I am William and I'm part of the wet lab team. So today I'll be going over all of our wet lab design and some future plans. So let us first introduce the system, which is a self-sustaining system. So it's similar to a cycle. And before we begin, let's just introduce our bacterial chassis. So we actually have two different strains that we're going to engineer for E. coli and cyanobacteria. So our E. coli strain is BL21BE3, which is an excellent candidate strain for protein recombinant expression. And our cyanobacteria strain is S. elongatus PCC7942, which is well studied and also naturally competent. So E. coli and S. elongatus cultures are in a symbiosis, where the cyanobacteria produces the sucrose for the E. coli, and the E. coli produces carbon dioxide for the cyanobacteria. At the same time, the E. coli can break down PET into, the E. coli can synthesize two enzymes to break down PET into monomers, two environmentally benign monomers. I'll elaborate on that later. So this is a self-repeating cycle where cyanobacteria and the E. coli are mutually beneficial in terms of carbon exchange, while the E. coli degrades the plastic. So this cycle repeats, as you can see, step 1 to 4 repeats. Yes. So let's go through the two main enzymes, PETase and MHETase. MHETase is also known as MHET hydrolase. So both enzymes digest the plastic through a series of reactions that involve hydrolysis. So here we can see the protein structure of the two enzymes, and they're actually highly related to its mechanisms. So we can see the protein mechanisms in action. So the PETase catalyzes the hydrolysis of PET into MHET, and MHETase then catalyzes MHET into two environmentally benign monomers. So in this case, it's terephthalic acid, TPA, and ethylene glycol, EG. So here's our plasmid design for the PETase digestion. So I'll briefly go over it. So this is for E. coli. And as you can see, like the vector backbone used is PSB1A3. It is a high copy number backbone with ampicillin resistance. And for the inserts, the most important two inserts, PET and MHETase, are both included, as I said before. So yeah. So next slide. I would like to just first clarify that only a few wild-type E. coli strains can use sucrose as a carbon source, such as E. coli W. So therefore, we will have to be able to engineer a plasmid for sucrose uptake. So here we will be incorporating sucrose utilizing genes. In this case, it's CSCA, CSCB, and CSCK. So CSCB acts as a sucrose permease that allows sucrose to be channeled in and out of the cell. And both CSCA and CSCK are genes responsible for metabolism of sucrose. So we incorporate these genes into the bacterium genome instead of merely transforming them as a plasmid. And we achieve this by using the lambda red system derived from a bacteriophage. So this ensures that the sucrose utilizing function will not be easily lost through the generations as the organisms propagate and carry out its cellular division. So after we establish this stable line of sucrose eating cells, we can then transform the plastic plasmid into these cells for the maximum efficiency. So yeah, here's our plasmid. For our sucrose plasmid, we're also using PSB1A3 as our backbone. And as you can see, the sucrose CSCA, CSCB, and CSCK are the inserts that will be incorporated into the bacterial genome to ensure the ingestion and metabolism of sucrose. And for the lambda red system, we will also be using a PKD46 helper plasmid, which contains the red genes for our lambda recombinatory. And last but not least, we're going to introduce our plasmid for the cyanobacteria, PCC7942. So let me introduce how to engineer our cyanobacteria to secrete sucrose. So the main gene involved is, of course, CSCB again, which is the sucrose permease. And this allows sucrose to be secreted outside the cyanobacterial cells in appropriate sodium chloride concentrations. And we also included the LAC-I gene, which codes for the LAC repressor protein. And we'll incorporate a gene for our IPTG inducible system. So this is our plasmid. So unlike our previous plasmids, this one is actually inside a low copy number backbone, PSB4A5. And this also has ampicillin resistance. So as you can see in the LAC-I and CSCB genes, they're both incorporated into this plasmid, and this will allow our transformed cyanobacteria to secrete sucrose more efficiently. Now we're going to go over some further plans for our engineering. So to achieve symbiosis, our future model will actually include a function for E. coli to convert atmospheric carbon dioxide into bicarbonate. So not only will this transform E. coli into a carbon sink, thus canceling out the effects of its own respiratory functions, It also provides readily usable bicarbonate for S. elongatus as an extra carbon source. So this optimizes sucrose production and thus E. coli function, achieving a true self-sustaining system and material recycling. And our second plan is to use symbiosis for another microbe, which can produce bioplastic. So in this case, it's Pseudomonas. So after digestion of PETase and AIGTase, to recycle the monomers, we can utilize the Pseudomonas strains from the Pseudomonas genus to their ability to produce bioplastics. So Pseudomonas strains can produce these things called polyhydroxyalkanoids, or PHA, as their source of energy. So PHA can be used instead of like the traditional textile PET as an environmentally friendly alternative. So with this, we can not only create something that's biologically recycling, but also turn it into a new product. So now I'll hand over back to Isabella to talk about the container design.
So we also have an initial design for the container, which consists of the external body, a pulverizer, and a transparent bioreactor. At the top, there's a pulverizer to grind the PET into smaller pieces and to increase the surface area for degradation, which will then fall into the center of the bioreactor containing E. coli and the enzymes. And inside, there is also compartments on either side for the cyanobacteria. So when sunlight enters the bioreactor, the cyanobacteria can carry out photosynthesis and produce sucrose, which can diffuse through the membrane separating the two organisms to reach the E. coli. Just to highlight why we chose this design, it's self-sustainable because the symbiosis allows it to only need the input of sunlight and power supply. And we also need to only increase the volume of bacterial medium to increase the level of degradation, making it easily scalable for larger operations. And as it's a modular design, we can easily replace or add parts in the future. Our project potentially brings some social and environmental impacts. Most importantly, we stop the creation of new PET monomers by introducing biorecycling of existing PET into new products. And in a similar way, it can also ease the environmental impact of fast fashion, which is a huge problem, and also for single-use food packaging. And last but not the least, companies can invest in our project if it takes off to offset their plastic production. So that wraps up our presentation. Thank you.
Thank you everyone for listening to our presentation.
Thank you Team Hong Kong HKU for this amazing presentation. We'll move on to the expert comment session. Know that we have a total 10 minutes for this session.
Perhaps I can I can start by asking a couple of questions here. So you have focused on using E. coli to produce PETase, and for PET degradation, PETase is an enzyme that works at room temperature, so I understand why you'd want to use it. But it is also not as efficient as cutinase, for example, or the LCC enzyme. But that one, of course, has the problem of only being really active at 70 degrees. So in terms of time scale, would this be a problem for your system, do you think? Because the PETase would be rather slow in degrading the plastic. This is my major concern. And and also one thing that was not included here, which I would like to point out, because maybe you mentioned it, I have to say my connection is a bit iffy sometimes. For this to work, the PETase has to be excreted. So you cannot have it in the E. coli, in the cytoplasm of E. coli. And this will not degrade the plastic, which is outside, of course. So it needs to be excreted. So you need to include the signal for excretion of the PETase. So I just wanted to know if you considered all of this. And another comment that I would like to say to make is that perhaps the system for the 7942, you may want to consider it as a constitutive system rather than an IPTG inducible system, because that will add to your cost of the whole system, right? And then finally, if you're planning to use Pseudomonas to produce PHA, then I would say you could use Pseudomonas to also produce the petase and mhetase, because then you would have only two systems to work with rather than three, which would, I would say, make it a little bit easier. In any case, I do like your idea. These are just suggestions for you to consider to perhaps improve it.
Thank you, Dr. Selão, for your suggestions. Yeah, thank you so much. We're actually considering to improve upon our container design. So the pulverizer definitely helps speeding up the digestion of PET. And for the Pseudomonas part, it's also in a preliminary stage, but it's also like a really good idea to incorporate plasmids that can actually produce PET in the Pseudomonas. So yeah, we might consider that. Thank you.
And to answer your question, like why we are using PETAs, we're using a combination of PETAs and metase. So I think it was more effective. And a research paper that we looked at was that PETAs was able to degrade 20% of plastics in the span of 24 hours, but that was for like modified PETAs. Yeah.
Yes, I mean, there's the fusion between the two actually that you can consider. This is a recent paper from Portsmouth, I think. So the fusion between petase and mhetase is actually beneficial. It will actually increase your activity more than just expressing the two. So this is something that you should also perhaps consider. Yeah, but in general, I think that it's an interesting idea. So you, of course, then you should hand over to him because you are trying to use 7942, which is a freshwater.
Did my screen lock or? Well, I guess I'll just sort of jump in here since first off, great presentation, a lot of fun. It's a really cool and a really big problem that you're trying to tackle. So it's an exciting project. I found one of the things that was sort of the most interesting and intriguing to me was the design that you had for the plastic degradation. Oh, well, I think you're back, right?
Yeah, now I'm back. Sorry, my connection is really awful. I'm in the countryside, so sorry about that.
I jumped in because I wasn't sure if you were cut off entirely, but please finish off or carry on.
Just asking, like, you were a marine strain so that you could target
Sorry, I think that's directed to me, but I can't hear anything. So something about.
You can just go ahead with your comments and
Okay.
we'll come back.
Okay. So I also had some questions just sort of about timing, right? Like, you know, and it might be fun to do some calculations, say for like a home user or whatever. It sort of looks like the way that you designed that system that was sort of almost like a washing machine style thing that was designed for in-home use. And so I was curious if you thought at all about that timing, like the question that we just got before, as well as if you've thought at all about how you're going to keep sort of the E. coli that are engineered in that container stable when you're constantly putting in, you know, plastic waste that might be contaminated with any number of, you know, bacteria. So those were a couple things that came to mind.
I do. So sorry. Sorry. May I go ahead? Yes. So just like a quick comment about the concerns you have raised. So our container design is quite preliminary, I would say. We haven't tested it out yet because the wet lab team is still focusing on the biological system. But currently we're targeting companies first. Yeah, not really for domestic use.
Okay. And I absolutely would not expect you to have actually built a prototype of that and conducted experiments or anything yet. But I guess kind of what we were both asking questions about is it might be interesting to think about what level or rate of degradation you would need in order to kind of have a realistic breakdown of a product over time. And you won't necessarily need to collect data. And this is a project that's much bigger than any one iGEM team, so you can't expect it to get there or anything. But it would be interesting to be able to present this is the rate of flux that you need, or in order to have a realistic breakdown on the order of days or weeks, as opposed to decades or longer.
I have a quick comment. Let me just.
Yes,
Okay, let me make a quick comment on something that I thought is very attractive in your biology scheme, which is this interdependence between at least the cyanobacteria and E. coli, because you are having E. coli make bicarbonate and cyanobacteria using it. Given that though, cyanobacteria, if it sees CO2, it's just going to use it. It's not going to depend on E. coli. So my question is, in your system, how are you going to make sure that the bicarbonate that's coming from E. coli is the only source of CO2 that cyanobacteria see? I'll be happy to elaborate more.
Sorry, is it about like the bicarbonate from the cyanobacteria?
Right.
Yeah.
Right.
It's the only source of CO2.
Right. So if the bicarbonate is from E. coli is not the only source of CO2, then in any kind of synthetic system where you have more than one organism, in this case, the cyanobacteria is just run off and leaving the E. coli behind, right? But you want to keep the two together, which is a very nice part of your project. So you may want to think about how not allowing cyanobacteria to grow with CO2 that's out in the atmosphere and depend exclusively on the E. coli CO2, which is converted to bicarbonate.
Again, I think this refers back to our container design as well as dry lab modeling. Since this is only like the bicarbonate production is only like a preliminary idea. So yes, it's a really good suggestion from Dr. Pakrasi, but we're still thinking about how this carbon sink idea can be optimized. Yes.
So we are kind of out of time, but if Dr. Taylor has any comments to make, quick comments, we can take it right now.
Okay, thank you. Well, it's a super important problem. Nice presentation. Best of luck. In your bioreactor, you mentioned that the only two things you need to add are CO2 and sunlight, but cells are more than just carbon. What about other nutrients, nitrogen, phosphorus, and so on? Have you considered how you're going to sustain these things with things other than plastic, which is only carbon, hydrogen, and oxygen?
We have considered that for our Cyanobacteria strain, which obviously grows in BG11 medium. That is quite I wouldn't say nutrient rich, but very trace mineral rich. And for that, we will, we will consider more for the E. coli part. So we understand this is some kind of bottleneck we're trying to solve, but yes, thank you for your comment. Yeah.
Okay.
Great. So I would request all the judges to fill out the judging form.
Also, in meantime, I request Team IISER-Pune to get ready for their presentation as they will be going next.
Are the judges done with filling the judging form?
I hope all the judges have filled the form. Thank you for your comments. Moving on to the next team. Team IISER-Pune-India will be speaking on sustainable biomanufacturing of industrial compounds using a co-culture of cyanobacteria in E. coli. I hereby request Team IISER-Pune to start presenting the project. They'll be having 10 minutes for this presentation.
Oh. Hello, am I audible?
Yes, Anjana, you're audible.
Hello? Audible? Okay, thank you. Let me just share my screen. Is my screen visible, Ravi? Okay, so hello, I am Sanjana from Team IISER-Pune, and this year we're working on recycling atmospheric carbon dioxide for biomanufacturing using synthetic biology. Carbon dioxide levels are off the charts, and we must bring them down to minimize the worst effects of climate change. Annually, around 43 billion tons of carbon dioxide are emitted into the atmosphere. Fossil fuels contribute to over 74% of the global emissions, out of which 18% of the industrial emissions come from chemical and petrochemical industries. India itself contributes up to 7% of the global emissions. Presenting our solution. So what do we want to achieve? We aim to create a carbon negative or carbon neutral model for industrial chemical synthesis to recycle atmospheric carbon dioxide and to generate renewable microbial feedstocks and convert those feedstocks into industrially relevant compounds. And how are we going to go about achieving this? We create a co-culture of cyanobacteria and E. coli to manufacture chemicals. Cyanobacteria fix atmospheric carbon dioxide as sucrose under osmotic stress. And we will modify it to secrete it into the culture medium. E. coli then takes up the sucrose and uses it to manufacture chemicals. The chemical we've chosen for this particular project is butanol, but by substituting the strain of E. coli we use, we can manufacture a virtually endless number of products. Our choice of strain is superior to plants on many different fronts. It thrives at high light, temperature, and CO2 concentrations, and is thus suitable for outdoor cultivation. It fixes a much greater proportion of its carbon as sucrose, 90% compared to 15%. It is also less resource intensive to grow and can subsist on waste such as industrial flue gas and wastewater, so it does not compete with food production and agriculture. The co-culture facilitates a single-step conversion of atmospheric carbon dioxide into the desired end product, and this end product can be varied by changing the heterotrophic strain used. There is no sucrose wastage as it is directly consumed by the E. coli and does not have to be manually extracted or transported. The cyanobacteria also generate toxic reactive oxygen species. E. coli can quench these, leading to better cyanobacterial growth. So moving on to the wet lab component of our project, we begin by modifying our cyanobacteria. They naturally accumulate sucrose under osmotic stress, so offers challenges to facilitate secretion. We intend to introduce a non-native sucrose transporter, CSCB. Additionally, to increase the yield of sucrose, we planned on overexpressing SPS, the enzyme that catalyzes the rate-limiting step of sucrose synthesis cyanobacteria. We wanted to match our yields to those reported in literature, and we also wanted to see if we could improve on these yields through our choice of promoter. We intended to test various strong native promoters, including some previously uncharacterized ones. We were initially planning to use commercial suicide vectors developed for BCC7942 as backbones for genomic integration of our genes into the neutral sites of UTX2973. However, as this strain is not naturally transformable, the plasmid would need to be introduced via conjugation with E. coli. Unfortunately, the suicide vectors we had access to have not previously been used in this way. The Cyanogate kit included suicide vectors compatible with conjugation, and we therefore decided to shift to it. This kit includes a library of parts of various levels flanked by standard restriction sites. We would clone our basic promoter, CSCB gene and terminator into a single cassette, level 1, and then clone that into a level 2 plasmid flanked by homology arms. Our mentors, however, advised us that this would be too complicated for the limited engineering we had in mind, especially if we wanted to build the promoter in our construct. We finally settled on a CRISPR-mediated integration to introduce our gene. We assemble our cassette through overlap PCR and insert it via KPN1 restriction cloning into the plasmid PSL2680. A gRNA specific to neutral site 1 is inserted via AAR1 and enables integration. The devastating second wave of COVID-19 in India delayed our return to campus by 4 months. Dr. Pakrasi has very generously agreed to lend us his strains of sucrose secreting Butex 2973. To characterize the promoters, we use a modified gene cassette with strong fluorescent protein instead of CSCB. To characterize promoter activity in this chassis, we vary light intensity and CO2 concentration and observe the strength of fluorescence in different conditions. The second step involves modifying the butanol-producing strain of E. coli lent to us by Dr. Yazani from ICGEB to take and break it down. Again, we start with the permease coded by CSCB, this time working in the opposite direction, and also add 2 additional genes, CSCA and CSCK, that incorporate all carbon sucrose into the native glycolytic path. We would like to thank Dr. Yazani. His advice on shifting to a microaerobic environment for our co-culture helped us select optimal growth conditions for our microbes. We are also indebted to Dr. Anama, who helped us reevaluate our assays and offered insights into the butanol game. Now we come to the computational aspects of our project. Our main goals in the dry lab this year include optimal strain design, which involves identifying key gene deletion or overexpression targets that could increase sucrose yields and important metabolic interactions between the 2 species in our co-culture, modeling the dynamics of the co-culture, how the biomass and key metabolite concentrations vary over time, and evaluating the impact of the biomass manufacturing procedure on the environment using methods like life cycle assessment or LCA. To identify the optimal modifications, we use constraint-based modeling techniques such as flux balance analysis, which primarily make use of genome-scale metabolic models. These allow us to optimize a particular objective, say growth rate, by manipulating the fluxes through the organism's metabolic network. A model for our cyanobacteria strain was provided by Dr. Costa. Since we had limited wet lab access, we had to make some assumptions on the uptake rates of certain compounds like carbon dioxide and to simulate salt stress. Growth rates and maximum sucrose production rates were the results we focused on at the start of our dry lab. Here we have 2 graphs from our preliminary rounds of the model. On the left is a biomass versus sucrose for different CO2 uptake rates, and it clearly tells us that the 2 are inversely related. On the right, we have biomass versus CO2 uptake rates at DB efficiency values. They're directly proportional to each other. Right now, we're working on finding more modifications using a particular extension of flux balance analysis, namely FSEOF. After applying the same techniques on E. coli, extend the FBA to a co-culture setting as follows. We assume that the 2 strains have equal time average growth rate handled by SteadyCom, and then look at permissible variability in metabolic fluxes at different values of this growth rate using flux variability analysis. Analysis of the tolerated variability of each flux helps us assess the importance of the corresponding metabolic interactions. If we incorporate the uptake kinetics of some of these crucial metabolites, we can predict the final yield and biomass values at different points in time through dynamic modeling. Finally, we would hope to plug in our optimal strains into the dynamic model to see if the effects of the overexpressions and deletions are significant in the co-culture setting by comparing the butanol yield. We'd like to acknowledge the help of Dr. Shri Srivastava, who introduced us to constraint-based modeling through flux balance analysis, and Dr. Dwarak Ravikumar, who helped us understand what a life cycle assessment can add to our project. Now moving on to our human practices, which we divide into 3 parts: collaboration, outreach, and integrated human practices. On the collaboration front, we've worked with 3 wonderful teams and organized the Cyanobacteria Symposium. We're also working with Team Toulouse to develop a dynamic metabolic models, and with another team to develop an international assess people's opinions on GMOs. To reach a wider audience, multiple activities like surveys, comic strips, webinars, explainer videos, etc., to introduce the general public to the world of synthetic biology. Coming to expert interactions for future implementations, we spoke to Dr. Shantanu, who helped us understand the importance of reusing waste biomass to make our project environmentally sustainable. Dr. Malti, on the other hand, told us about the limitations of using biomass as animal feed. We also spoke to Dr. Anand, who helped us identify potential stakeholders and downstream processing strategies, and Dr. Reena Pandit, who told us about parameters to consider for bioreactor design. To conclude, we would like to thank our PI, Professor Thomas Fukudil, and our advisors, Prem, Viral, and Chinmay. We'd finally like to thank all our sponsors. Thank you. And we're now open to any questions. Thank you for listening.
Thank you, Team IISER-Pune. I now request our judges to give their input.
Could I ask just a real simple question? I may have missed it in your presentation, which was very clear. What are the applications for the butanol? How will that be used?
We intend for the butanol to be used as a biofuel or a solvent or any other common chemical uses that it has.
I guess my concern is there, it's kind of circular. If you're going to use it as a biofuel, then you're just returning the CO2 back to the atmosphere.
Yeah. So the aspect of whether it is carbon negative or carbon neutral would depend on how the carbon is sequestered. So with this proof of concept of butanol, as if it's being burnt, it would release this CO2 back into the air. But again, the CO2 is not coming from the ground. It is atmospheric CO2 that is being recycled. And we hope that we get, like, our project is more sustainable because we are diverting or using carbon which is not in the ground, but instead using carbon which is from the atmosphere.
So in effect, it's neutral. It's a carbon neutral solution. Okay, thank you.
Yeah, and to add to that, if we take on more compounds in the future, we would hope to create a carbon negative process this time. For butanol, it's going to be a carbon neutral process.
Okay, thank you.
I have a question, a similar question about what Dr. Taylor just mentioned to you. That's something important to keep in mind. There is really no reason to constrain yourself to biofuels, right? You are developing a chassis or a system that can produce many different components. And these days, people are thinking quite a bit about the other side of the story, which is food and nutrition. So have you thought about anything regarding that? And how would you approach that?
In our bioreactor design, we have been talking to experts about how we could try and recover the waste biomass. And some of the inputs we had received was using that biomass as animal feed. And with regard to that, we have decisions to make about the safety of that biomass, whether it be suitable for animal feed or human consumption, especially since it's genetically modified and if it has an antibiotic marker or anything like that.
And to answer the other part of your question, so initially we were trying to focus on succinic acid, which had a pretty large role to play in the food industry. And due to lack of time, we were offered a butanol producing strain, which is why we tried to switch to it. But in the future, when we scale up our project, we're thinking of developing it further. We will definitely consider a wider array of products and address products that really have an impact on the society.
Good.
Great presentation and a really exciting and fun project. I'm going to focus just a little bit in the questions on the specific project or product that you chose. And I was curious if you had talked much about how you're going to potentially measure butanol. And I was also really excited about the metabolic modeling and sort of co-cultures that you're doing. And I was curious if you're able to, well, one, sort of from the literature predict if there's going to be an impact of butanol on either of the species in this co-culture, and if you would include sort of any effects potentially of butanol or toxicity of butanol as part of your modeling.
So we just got wet lab access. So we're trying to perform MIC tests to figure out the tolerance of E. coli and cyanobacteria to the butanol. And once we get those values, we'll incorporate it into the model and figure out what impact it has on the co-culture and the stability of the co-culture.
Great.
Just to add to that, we'll also be, so we're looking at an integrated bioreactor design where we want to remove butanol in situ as opposed to how ethanol setups work. So that in itself should pretty much remove any toxicity effects because the media is constantly being changed in the bioreactor setup.
Do any of the other experts have any questions?
I do not have any I do not have any question, but something that I wanted to mention to all iGEM team members. We have been in communication, my lab has been in communication with the IISER-Pune team, and I know how difficult these days it has been to receive strains from different countries. Don't worry about it, we will send the stuff again by FedEx, but I'm sure that everybody is suffering from the same issue of transferring material from one country to another country. So it's a challenging time.
Thank you so much. We're really grateful to you for offering your, for accepting our request in the first place. It really cut down on our project time by a lot, and we're really grateful to you and your lab for helping us out in these times. Thank you so much.
Great. So if there are no further comments, I guess we can move on to filling the judging form. We have 5 minutes.
I'd also like to request Team Toulouse to start preparing for their presentations as they'll be presenting next. Hello, judges. I'd like to apologize that there's been slight misunderstanding on our side. IISER-Pune had 4 minutes left for their judging session, and they had some questions for all the judges. So I think, so can the judges take these questions?
Yeah, of course.
So one team member has put a question in the chat box. Maybe I'll read it out loud. If I may ask the experts here, would you recommend modifying cyanobacteria through genomic integration as opposed to transforming them with special self-replicated plasmid? Are there merits or demerits to both?
Himadri, do you want to handle this since you do more UTEX?
No, go ahead, Danny. Go ahead, Danny.
Well, I'm less familiar with the particular strain that you're using. I would say that genomic integration is probably it's definitely preferred for my organism because it's just more straightforward and simple. And some of the replicative plasmids are not as reliable, and you always have the problem of potentially losing the plasmid over time. But I've not ever really directly done a transformation of UTEX. So that's why I was going to say, Himadri, if you want to chime in.
Okay, so it depends on what you want to do. Both genomic integration and trans expression in self-replicating plasmid are eminently doable in UTEX 2973. It depends on the demand of your experiment. Typically, what one does in not just in cyanobacteria, but any production system that you are setting up is try it quickly by using self-replicating plasmids, because you can have many combinations and very quickly go and test it. Once you have the gene expressing in the organism and you figure out some of the nuances, then you want to have it integrated into the genome, as Danny just pointed out, because in a big production system, the worst possible thing that can happen, and it has happened in the past with other bacterial systems, is that you lose the plasmid. And if that happens, then there is no production. So genomic integration is very good. The other thing that you need to consider is the level of expression. Oftentimes, people use self-replicating plasmids that have higher or lower amount or a higher number of ploidy. So if you want to produce something in a very large amount, an easy way to do that is to put it in a self-replicating plasmid that's present in a high copy number. Okay, so it depends on what your experimental needs and demands are. I see the other question about antibiotic markers. There is no need to use antibiotic markers anymore. Just go for deletion constructions. If you are trying to get rid of things, use CRISPR, because antibiotic resistance markers, as has been pointed out in the past, are going to lead to all kinds of downstream problems. However, if you have any issue with the with ploidy levels, meaning that you have wild type copies are still present, then antibiotic resistance usually gives you a leg up.
Yeah, I would just, as a matter of practicality for, especially projects with short time windows, say that, the antibiotic approaches, we all know it has problems in scale up, but it's a straightforward way to sort of get quickly to the strain that you want, kind of analogously to the plasmid versus genomic integration, a question that Himadri just sort of said.
Just to clarify, Dr. Pakrasi, are you implying that when we use antibiotic markers, we select for those bacteria which have a higher copy number of the integrated gene? And that's why it works when we are aiming for higher ploidy?
No, that's not the question. If you have, so as you know, cyanobacterial genomes, and not just cyanobacteria, all bacteria, they can have different ploidy levels of their genome, genomic DNA. So a challenge has been around for the longest time that you have both wild type copy and the mutated copy are present in the same cell, and you have no way of knowing that. So people usually use antibiotic resistance and then subculture over and over again so that you can get rid of all the wild type copies. The good thing about CRISPR is because you are making double cuts, you are going to lose majority of your chromosomes except the ones where you have repair to put your DNA in. So it goes again in both ways, but the antibiotic resistance offers you a quick and easy approach. That has nothing to do with ploidy. That was more about the plasmid.
I see.
Thank you so much for your inputs, and I apologize once again for the misunderstanding on my side. I request the judges to resume filling the judging form.
Once again, Team Toulouse, do prepare for the next presentation.
Yeah, you will be presenting in next 5 minutes.
Do we have to present it now or in 5 minutes? Sorry, I didn't understand.
Yeah, in 5 minutes. We'll let you know when to start.
Okay.
I hope all the judges are done with filling the form. Great, so let's move on to the next team. So the next team is Team Toulouse INSA-UPS, who will be working on synthetic microbial consortium for sustainable production of violet scent molecules. You may go ahead and present. You have 10 minutes.
Okay, thank you. So hello, everybody. I'm Margot, and with my teammate Thomas, we will introduce you our project called Elixio, which aims to give back its voice to the violet. So first, before explaining you our project, I have to tell you more about mute flowers. Mute flowers are flowers from which it is impossible to extract the fragrance, regardless the process used. And this phenomenon concerns a lot of flowers in which the violets, however, the scents of this flower are really sought by industrials, and nowadays these scented molecules, are manufactured using fossil resources. So these molecules are not sustainable and cannot be considered natural.
With Elixir, we want to give back its voice to the violet, and our solution is to develop a platform for violet flower accord through synthetic biology. In order to achieve our goal, which is to create a modular platform for the synthesis of the first natural violet flowers accord into a yeast, we have to reach a lot of steps in which we want to link synthetic biology to the art of perfume creation. We also want to make a sustainable and biosourced accord, and we want it to be labeled as natural and sustainable. And of course, we want to create an accord that mimics the smell of the real flowers by producing the molecules present in the flower naturally. And then, sorry, Thomas. And then we want to collaborate with perfume makers to find interesting fragrance mixes. And the expertise of these perfume makers will lead us to the perfect violet accord for our prospects. I will now let the floor to Thomas, who will explain you more about the scientific aspects of our project.
Okay, so hi everyone. So the main flower volatile molecules are 6 molecules that we choose. It consists of 4 terpenes, the alpha ion, the beta ion, the dihydro-beta ion, and the linalol, and 2 other molecules which are aldehydes. So this is the 2,6-nonadienal and the 2,6-nonadienol. So we aim to produce them with a synthetic microbial consortium between yeast, so the Saccharomyces cerevisiae strain, and the cyanobacteria, which is the Synechococcus elongatus, because we want to make express the violet terpenes with the Saccharomyces cerevisiae strain and the aldehydes with the Synechococcus elongatus strain. And so we've seen that these two, there is one heterotrophic module and so an autotrophic module. So we imagine that theoretically we can make a bioconsortium between those two organisms. So for the more metabolic engineer aspect of our project, we want to start with the Saccharomyces cerevisiae strain. So we need to produce them the violet terpenes. And to produce those terpenes, we need a metabolic pathway called the mevalent pathway for Henberg pathway, which is already engineered in a modified strain that we have in the lab. So the mevalent pathway can be divided in 2 steps. So the first step is the upstream pathway, where the IPP and the DMAP, the 2 main precursors, are produced. And in the downstream pathway, those 2 precursors are condensed to produce 4 families of terpenoids. So there is the monotapenoids, which are a molecule, 10 carbon-based molecule. There is the sesquiterpenoids for the 15 carbon molecules, the diterpenoids for 20 carbon molecules, and so the triterpenoids for 30 carbon molecules. So as explained before, the molecules I want to produce is the linalol. So this is a monotapenoid, and so we have to only clone one-step reaction with an enzyme called the linalol synthase, so for this. And the 3 other molecules are triterpenoids. So for the alpha ion and the beta ion, we use as precursors the lycopene, and we use 2 enzymatic fusion with the CCD1. So for alpha ion, we have enzymatic fusion between LCYE and CCD1 to produce alpha ion. And for beta ion, we use an enzymatic fusion between 30Y and CCD1. And so to obtain a dihydro-beta ion, this is only a 1 step before beta ion with the enzyme DRB1, which will be cloned into the yeast. So now for the metabolic engineering of cyanobacteria, we want to produce, so I said, the 2,6-nonadienal and the 2,6-nonadienol, and there is, I have as precursor 2 well-known fatty acids, which are the linoleic acid and the linolenic acid. And so we have to clone 2 enzymes. This is so the 9-LOX and the 9-HPL to obtain the 3,6-nonadienal as precursor. And since the Cyanobacteria Synechococcus elongatus has already an IDH enzyme, we can have so the 2,6 non-diurnal and the 2,6 non-diurnal theoretically. So as explained, for this was the most metabolic engineering side of our project, but we focus on two the co-culture and the ways that we want to extract the molecule sent in the media of culture. So for the co-culture, as I said, we use Synechococcus elongatus strain from the Pakrasi lab, which is a CSB plus phenotype. So it can produce, it can excrete sucrose in the media of culture, and the yeast strain that we use can metabolize as carbon source the sucrose, and so we can imagine a co-culture between those two microorganisms. Since we imagine that we can recover the molecule from the media of culture, we have seen that there is we can make more sustainable process to extract this molecule, because nowadays use methods relies on sometimes organic solvents, so with petrosourced solvents. And so we found that there is a new way to extract this molecule called the evaporation, which use a membrane that extracts selectively our molecules of interest from the media of culture without recovering the GMOs into the media of culture. So we can, with this process, extract specifically our molecules of interest. So thank you for your attention.
Thank you, Team Toulouse. This is a really nice presentation. We can now move on to the expert comment section.
Great presentation and really creative and fun project. One thing I really love about this project is that you're using both species to sort of produce one component of the end product. And I think that that's novel and an interesting approach and kind of using the strengths of both species. But it does also raise the question of then how do you think about balancing the product output from one species to the other? Like, you know, if your yeast are depending mostly on the cyano for sucrose, but that they're, you know, there's one flux there that needs to be balanced, but then maybe their production rates for the terpenes versus the aldehydes are very different. And so how might you go about making sure that it doesn't smell overly floral or too leafy?
Yeah, we think that there might be a limitation of the excretion of sucrose in media culture, since we make a novel engineer into the cyanobacteria, but we don't even now try to make the co-culture with the yeast. So maybe our modeling team is trying to solve the problem, but we don't have now imagine a way to make a stronger expression of sucrose in the media culture. Yeah, you're right.
So I have a question regarding modeling. I was waiting to hear from you what kind of specific modeling activity you are undertaking. Maybe you have mentioned this, but it missed my attention.
With the modeling part, we try to know what flux are produced of our scented molecules. And as we said in the presentation, we want to make a modular accord. So we want to know if I'm sorry, it's difficult in English. Maybe with some other sources of carbons, maybe we could have different amounts of these scented molecules. I don't know if I was very clear.
Yeah, well, thanks for the clarification. And this is exactly what I wanted to point out. In order for the modeling to go hand in hand with your wet lab experiments. This amount is a good word. So now for the wet lab part of the study, are you planning to measure the amounts of the different compounds that are being produced? Because for modeling, you need that, right?
Yeah, absolutely. We talked to some team with some lab team with work on the identifications and the quantification of those molecules. And we see that with the LCMS methods, we can measure the quantity of those molecules of interest that will be produced in the media of culture. So yes, we can follow the production of those molecules.
And with HPH too.
That's
nice. Okay.
Yeah, nice presentation, guys. In the extraction process, are you extracting both the terpenes and the aldehydes from a single suspension, or is that done separately in the co-culture?
No, there is. I see what you mean.
Yeah.
There will be more than one membrane of propylphenol because as I said, yes, the membrane is specific for one family or one specific molecule. So as the aldehydes and terpenoids are different, we need maybe more than we will have to find more than one membrane to extract these two families of molecules.
Thank you.
Are there any questions that you would like to ask to our experts?
We still have 5 minutes for your judging session.
Okay, so let's think about the downstream processes. And I'm asking these questions because I am not quite familiar with. So we have been talking about different kinds of compounds. We're talking about biofuels. We have talked about other things with fragrance. And it's interesting that it's coming from your university, and the area probably produces a lot of these compounds. Do you have any idea about the market size about these compounds?
Yes, the beta ionon for the market of the beta ionon, it is very wide, but it is not. The beta ionon is not only used for perfume. It is also used for food and beverages, for example, and maybe in some case for medicine. So with our projects, we can step up from our competitors by making an accord, and also for the perfume industry. So I think we can overtake the market thanks to our differences.
That's good to know.
Is there anything that's particularly sort of problematic about the way that these synthetic fragrances are currently made?
Yes, so as I said, the violet is mute flowers, so we cannot take the scent from the flower itself. So in order to respond to the market demands, these molecules are produced with fossil resources by chemical processes. So it takes a huge amount of energy and of course resources. So with synthetic biology, we aim to develop a more sustainable perfume industry.
I really love that the sort of tagline of giving the voice back to the violet, and I can get behind this project. But one thing you might want to highlight more is sort of what those current costs are, or what makes it particularly attractive, if there are drawbacks to that current synthetic way of producing the violet fragrance.
Yes.
Yeah.
Yes, that's a very important thing. And for example, for chemical production, is it very easy to make only alpha ionon versus beta ionon? Does biology offer any advantage there?
Yes, chemically, I don't know how to say it. We produce beta ionon and also the chemically opposed molecule. Is it? However, with synthetic biology, we produce only beta ionon, and so we will afford to extract only one molecule and not the other one. I don't know if I'm clear here. Maybe Thomas.
Yeah, this is a no. Your answer is fine. Time is limited, but your project is very exciting, as you can see from Danny's dog.
Yeah.
Yeah,
And anecdotally, one of my favorite breath mints that's made here in New York is called Violette. And they're delicious. And I thought that they were getting the essence of that violet flavor from the flower itself, but now I know that's not true.
Yeah, but it's more complicated because maybe you use some absolute leaf violet because there is some perfume that is made from more the violet leaf and not the flower. So they say that it is violet, but it's more the leaf of the violet. So, and there is some culture of the leaf, but there is the same problem for the leaf of the violet because there is some monoculture of the violet, and it is a really little flower, and it is really hard to cultivate them. So yeah, for so many reasons, it is really complicated to cultivate the mute flowers. So yeah.
Okay.
Thank you so much for your inputs. I request all the judges to start filling the judging form. Also, next up is Team Bono. I request all the team members to get ready for the presentation. Thank you so much, judges. Up next is Team Berno, who will be speaking on harvesting phosphates from water using polyphosphokinase entrapped inside bacterial microcompartments produced by engineered Bacillus subtilis. I request Team Berno to start their presentations. You'll be having 10 minutes to talk.
Hi, I will share my screen now. So can everyone see my screen?
Yes,
I guess so. Okay, so hello everyone, my name is Klára Odehnalova and I would like to introduce our project, which is called PhosCage. So it is estimated that half of the world's food supplies rely on phosphate fertilizers. And while their natural reserves are predicted to be depleted within the next 80 years, phosphates are often used very inefficiently in agriculture. And as a result, a large portion of these fertilizers end up in surface waters. Obviously, high levels of phosphates in rivers and lakes can speed up the process of water eutrophication and thus lead to the overgrowth of cyanobacteria. This obviously poses a health risk for people and disrupts the balance of local ecosystems. So in our project, we would like to focus on developing a device that uses engineered bacteria to sequester phosphate from polluted waters and to recycle it as plant fertilizer. So first, I will introduce some theory to you. Gram-negative bacteria typically metabolize phosphates by using the enzyme polyphosphate kinase, which link phosphates into polyphosphates. These polymeric structures are then stored in cytoplasm, where they can be again broken down by exopolyphosphatases and used by the bacteria. Gram-positive bacteria, such as Bacillus subtilis, which we will be using for our project, incorporate phosphates into teichoic acids, which are then used as a part of their cell wall and are stored in this way. Obviously, they can also be broken down again. So next, I would like to introduce BMCs, or bacterial microcompartments. These are large hollow protein complexes, which can enclose certain biochemical reactions and are created by some bacterial species. Inside these BMCs are localized certain enzymes, which catalyze this biochemical reaction, and they kind of close off this reaction and separate it from cytoplasm, which allows for a special environment to be created, which is suitable for the reaction while not necessarily being suitable for the bacteria itself. The products of this reaction might also be too large to pass through the pores of the BMC, which then protects it from certain enzymes in the cytoplasm, which might digest the product. So in our project, we will first utilize an intake system for phosphates, which Bacillus obviously already has, as it needs these phosphates. And we will then modify Bacillus subtilis to produce these BMCs, inside of which we will enclose the enzyme polyphosphate kinase that we've previously talked about. This enzyme will obviously then polymerize phosphates into polyphosphates. Encapsulation of polyphosphates into BMCs should prevent their remetabolization and increase the storage capacity of these bacteria. So now we kind of have the basic gist of our project. However, there are some issues that came up. Firstly, BMC production can be quite straining for the bacteria, as it does require a production of large number of different proteins. So ideally, this process should not be happening constitutively and should be induced in some way. Also, another issue that should obviously be at the forefront of any project tackling genetically modified organisms is how do we prevent GMOs from getting released into water while obviously wanting to clean natural waters of excessive phosphates. So now let's talk about how we chose to tackle these issues. So in order to control the BMC production, it obviously is kind of intuitive to use the increase in phosphate levels to induce BMC production. So how could that be done? Most bacteria obviously need some way to control their phosphate intake. Bacillus subtilis and many other bacteria use the PAW pathway for this reason. It is a way for bacteria to respond to phosphate concentrations, and it consists of the PAW receptor, which is activated by low phosphate concentrations, which then activates the signaling molecule, which then turns on the PAW promoter, which obviously triggers the expression of many different genes regulating phosphate intake. What we've done is we've taken the PAW promoter, which gets activated by the phosphate pathway and which is already present in Bacillus subtilis, and placed it in front of a gene encoding a repressor. When expressed, this repressor will stop the production of BMCs and thus kind of give our bacteria the space to grow and thrive without needing to burden themselves with BMC production. However, when there is a higher phosphate concentration in the environment, the phosphate receptor will get turned off, and obviously the entire pathway will not be activated, and thus the repressor is not being produced. And as we've labeled the repressor molecules with degradation tag, the repressor, which is already present in the cytoplasm, will then promptly get degraded, and our BMCs will be free to be expressed. So, yes. Another question, obviously, is how do we make sure that our bacteria do not escape into nature? So first, I would like to introduce biofilm to you. Biofilm is a structure which is created by some bacteria by producing polysaccharides and proteins and excreting them into their environment. It is mainly used for adherence, communication, and protection. What we would like to do is mix our genetically modified Bacillus subtilis with a native strain of Bacillus, which is capable of biofilm production. So our modified bacteria will benefit from being placed inside of biofilm, which is obviously generally quite beneficial for the bacteria. But it will not need to burden itself with its production, as the native strain will take care of that. Biofilms can also be 3D printed into a suitable shape. And from what we found out, they should generally be safe to be used as a part of fertilizer as long as all the bacteria inside is no longer alive. So how would our project look in the real world? We still have some questions and uncertainties. So this is only kind of like a basic gist of what we're thinking about. Originally, we wanted to place our 3D printed biofilm into a floating device, which would obviously require very high standard of protection from GMO release, which could be ensured by coating biofilm in hydrogel, which is a substance that could also be 3D printed with our biofilm and which could ensure that no bacteria escape it. We are, however, still not sure how that would impact the vitality of our bacteria and how that would also kind of affect how the phosphates can get to our bacteria. Another thing that we are considering is placing our biofilm in wastewater treatment plants. We would also like to 3D print our biofilm into a shape which would maximize its surface area, which would obviously increase the efficiency greatly. So what makes our solution unique? By combining BMC production with phosphate biosensor, we will increase the fitness of modified bacteria and thus create an organism which is capable of efficiently producing BMCs without the need of outside induction. Our project will aim to combat cyanobacteria overabundance while also creating a creative way of recycling phosphates from agriculture and using them in fertilizers. So now I would like to thank our sponsors as well as you for listening to my presentation. And now I welcome your questions and feedback. Thank you.
Thank you, Team Bono, for your presentation. I now request the judges to give their expert advice.
Okay, could I start? Very nice presentation. I may have missed it early on, but why did you opt not to use gram negatives that produce copious amounts of polyphosphate already?
Well, first off, I guess we've had previous experiences working with Bacillus subtilis, and also this bacteria is very good at like surviving at lower temperatures that would be in the natural waters if we chose to place it there. So that was one of the reasons, I suppose.
Okay, all right. You touched on GMO release in the environment, and of course that's a big concern. In your schematic of the field application, you're talking about incorporating them into hydrogels and passing water through them. Out in the real world, water isn't just water. There's all sorts of debris and destructive things that would probably ablate, erode, and destroy your hydrogels as the water moves through your system. So that you might want to rethink that application.
Well, we would like to kind of 3D print it into like a shape with like pores in it or something like that to increase the surface area, so the water wouldn't be passing through like a block of hydrogel and biofilm. That wasn't really the plan, but
Okay.
Obviously that's kind of the thing that we don't really know yet, how well the hydrogel and biofilm would hold up in a stream of water, and that is something that we still kind of need to think on. So I do know that like that might not be ideal, and we are kind of considering our options on that.
Yeah, that's way down the line compared to what you have to do upstream. Yeah. Very interesting. Thank you.
Hello, if I may say something to that. Also, our IT guys that are working on the device and that worked on the device also for our project that we had previous year, they are thinking about some filters that would be like in front of the biofilm and hydrogel that would prevent the debris from coming into this part with bacteria.
Yeah, a great project, really clearly presented, and it has a lot of like cool components to it, and really great graphics too for whoever your designer is there. I was curious about the use of BMCs, and I don't know if you mentioned a specific type that you were planning on using, but I suppose maybe a beginning question would just be if you could elaborate slightly on why put your polyphosphate into a BMC in the first place.
So like obviously the polyphosphates could then be, if they are just in the cytoplasm, they can be like remetabolized by the bacteria and again broken down into phosphates, which might decrease the like efficiency of the phosphate accumulation, which we are hoping to prevent. We obviously don't know if that will happen. We obviously want to test it and see, and it might be like a good way of like increasing the efficiency of the process, which we like need to find out. And about the specific type, we will be using like the PDU operon, and from like a specific type of bacteria which is quite close to Bacillus subtilis, so it should work, hopefully. I do not remember the name right now, but
I'm sure you've talked about this, but part of the reason why I asked that question, right, is that there's a bunch of additional just biological almost technical considerations when you're thinking about making the BMC, like how is it formed? Is it forming from the outside in or the inside out? I think with a PDU compartment, you can expect it to form all on its own, but then you have this question of sort of transport of phosphate across the shell and how you're going to localize your enzymes into the BMC. And all those are really cool, exciting, interesting questions, and probably more than we really have time to get into just now. But you might get some questions about that strategy for how you're going to target your enzymes and the polyphosphate specifically there. I wondered, sort of to play devil's advocate there, if it would be possible to just knock out the genes that are responsible for utilization of polyphosphate, and that would, still create a cell that's going to be enriched for polyphosphate, and without having to go to the additional trouble of putting it into a BMC.
Well, obviously, though, these genes are still kind of tied into a lot of vital processes of the bacteria. So like we couldn't really knock them out entirely. Maybe we could like repress them by some obviously like way of inducing, I don't know. But that would obviously need a lot more research into all the metabolic pathway that this is tied into, which obviously is like ITP production and stuff like that. So that would probably kill the bacteria, I guess. But like for some time it might work. Like it's an interesting idea, thank you.
Are there any more comments from the judges, or are there any questions from the team?
I'll just elaborate on that last comment, which is to say that, I think it's a really neat concept. Just you might want to prepare some response to a type of question like the one I just posed, of why the BMC specifically, and what would the alternatives be.
Yeah, sure, thank you. And just like a quick comment, like you mentioned the stuff like how do we localize the enzyme and stuff like that, like we have that figured out. It just didn't really fit into the time slot of the presentation. So like if you'd like, I could like talk about that, but I don't know if that's very interesting really.
Sure, talk a little bit about it, that'd be great.
Yeah, so basically there is a study that uses this particular type of BMCs, and while they only enclosed GFP in it, they have managed to do that and confirm that that is happening. And they are basically using a part of one of the PDU proteins, which is like on the inside part of the BMCs. And it is the part that interacts with its neighboring proteins basically. And they have kind of decreased the size of it so that it can be used as a localization tag for another enzyme. So we will be using that. And yeah, the way that the BMCs kind of like are put together should happen on its own. So yeah, we are hoping that it will.
Is this the synthetic BMC that people were using SpyTag, SpyCatcher to localize enzymes to the lumen of the if so, that might be one way to something you could highlight on your cartoon diagrams in your presentation.
In this area is when you get the approval for a fund.
Sorry?
For a fund that you.
Sorry, I didn't quite catch that.
You're muted.
One other thing to maybe talk about briefly, if you have some time that you can squeeze it into the presentation, is just what your thoughts are about how you might get the phosphate to transport across, once you've actually assembled the BMC and presumably it's stable and fully formed, if there's a pore or a type of shell protein that you're going to engineer to allow that really highly charged phosphate group to go across.
Like the phosphates, they should pass by themselves through the pores of the BMCs, but you are right that like one way to really increase the efficiency of the process might be to incorporate like a transport molecule into the BMC. That is obviously probably too complex for this year's project, but is a really good idea for the future.
If you know that phosphate passes already, that's great. You might just want to mention that or know which shell pore that it transports through, because all of these different shell proteins have like a different sort of pore size and a different charge to their central center of the hexamer that then is going to influence how permeable they are or not to those kinds of small molecules.
Great. So we are out of time for the comments, but I request all judges to fill their forms. We have 5 minutes for that.
Okay. So thank you for your questions and the feedback.
Also, I request Team Linkoping to get ready. They will be presenting next.
Should we present now? Sure.
In 5 minutes, once the judges are done filling the form.
Okay, thank you.
I hope the judges are done with filling the form. Great. So coming up next is Team Lycopene, who will be speaking on a synthetic biology approach to desalinating seawater. You may go ahead with your presentation.
Thank you. So, can you hear me? Okay, so hello everyone. Welcome to our presentation. My name is Alexandra.
And my name is also Alexandra.
And we're from the Linköping iGEM team from Sweden.
We are very happy to be part of the Cyanobacteria Symposium and excited to present our project to you today.
So in this presentation, we will start by giving you some background as to why we chose our project. Then we will walk you through the basics of our project and all the different parts and how they come together to create one working product.
But let's start with some background to our project. Today, we are almost 7.9 billion people sharing this planet. The population is constantly increasing, leading to the expansion of integrated agriculture. As a result, the need for fresh water is greater than ever, and it will keep expanding. But despite the increasing need for fresh water, the water assets we have at our disposal are limited.
70% of the world's surface is covered in water. However, only 2.5% of that is fresh water. And of that, as little as 1% is available to us, whereas the remaining amount is bound in glaciers and snowfields.
As of today, the effects of freshwater shortage are already apparent in all parts of the world. It's vastly affecting the agriculture prospects in developing countries. But even here in Sweden, the agricultural industry is experiencing hardships caused by the lack of fresh water. It's something that will continue to have a major impact on the agricultural industry and the everyday life of the people relying on agricultural resources.
So to manage this crisis, various methods for desalinating seawater have been developed. And today, many industries and areas are dependent on those methods for their freshwater source. However, while these methods have found clever ways to utilize seawater to create fresh water, they have some apparent drawbacks. They're highly energy consuming, and the energy they use is often not renewable, causing them to leave an environmental footprint. The high energy consumption also makes them very costly, leading to them only being accessible to richer countries and industries.
So to offer a more environmentally friendly and economically accessible solution, we would like to present our project, Sea Salt. Sea Salt is a novel synthetic biology solution to the freshwater shortage. Our aim is to desalinate seawater using modified cyanobacteria to create fresh water for agricultural use.
Our desalination solution follows what we have chosen to call the sea salt cycle. The sea salt cycle shows the four main phases of our method, starting with the cultivation of the modified cyanobacteria, moving on to the actual desalination process, and then the separation of the cyanobacteria from the desalinated water. The cycle ends with part of the bacterial biomass being used for inoculation of the preceding cultivation, and the rest being used for biofuel production.
But let's start with taking a closer look at the first phase, decultivation. As we already mentioned, we will be using cyanobacteria for our project. More specifically, the Cyanobacteria Synecocystis sp. PCC6803. However, during the rest of this presentation, we will refer to it as Synecocystis to make it a little bit easier to follow.
Yes, and the main reason for us using Synecocystis is that they are both halophilic and photosynthetic. So what this means is that they can grow at high salt concentrations and that they utilize sunlight as their primary energy source. And due to these properties, both cultivation and desalination can take place at seawater salinity levels, and they can be driven by sunlight.
And after cultivating these modified Synecocystis, it's time to introduce them to the seawater so that they can desalinate it.
And what we mean by desalinating seawater is that the extracellular sodium chloride is transported into the Synecocystis cells and thereby removed from the surrounding water.
So let's explain how the desalination is achieved.
Well, It's made possible by a number of modifications done to the Synechocystis. First of all, we will genetically modify Synechocystis to express the inward-directed chloride pump halorhodopsin originating from the archaea Natronomonas feronis. Halorhodopsin is a light-activated ion pump, and when activated by light, it transports chloride ions into the cells against the concentration gradients.
And this influx of negatively charged chloride ions creates a negative membrane potential, which is expected to cause an influx of cations. To help the influx of sodium ions along, we will also make Synechocystis express an excess number of one of their native ion channels, MsCl. This way, the positively charged sodium ions can move into the cells via MsCl.
Through these modifications, the extracellular chloride and sodium concentrations will be decreased, meaning that the salinity of the surrounding water will be lowered.
The next step in the sea salt cycle is to separate the Synechocystis from the desalinated water. This will be achieved by yet another modification done to the Synechocystis.
And this modification entails making the Synechocystis express a fusion protein on their surface. The membrane-bound part of the fusion protein is an S-layer protein called SLR1272 that is endogenous in Synechocystis. The other part of the fusion protein is a cellulose binding domain, or CBD for short, originating from Clostridium thermocellum.
CBD has a very high affinity towards cellulose, which will allow us to filter out the Synechocystis from the desalinated water by passing it through a cellulose filter. The CBD on the bacterial surface will bind to the cellulose in the filter, and the Synechocystis can thereby be separated from the water. The filtered desalinated water is now ready to be used in agriculture.
So now we can move on to the last part of the sea salt cycle, because in addition to receiving desalinated water, we'll also make use of the leftover Synechocystis biomass that is stuck to the filter. Part of it will be used for inoculation of future Synechocystis cultures, and the rest can be used for biofuel production. And this completes the sea salt cycle.
And as you understand by now, the protagonist of the sea salt cycle is the modified Synechocystis. But despite all the advantages of using Synechocystis for this project, one thing worth mentioning is that Synechocystis has a slow growth rate compared to many other bacteria, such as E. coli. So to work around this issue, we are also using the fast-growing and halophilic bacteria, Vibrio natrigens, to confirm expression and positioning of the expressed proteins.
And to ensure that the expression is successful, we will validate all the modified parts in both Synechocystis and Vibrio. And we will do this by attaching the fluorescent protein GFP to our biobricks. And this way, we will be able to visually confirm expression and also see where in the cells our proteins are expressed.
Then, to confirm that our desalination modifications function as we want them to, we will measure the changes in extracellular and intracellular sodium and chloride concentration. We will also measure the biomass before and after filtration to see if our separation system works as anticipated.
But we wouldn't have been able to do all of this without help. There's a whole network of people that have helped our project so far.
First of all, from people in the academic world, we have gotten advice regarding working with Synechocystis, which is something we had no prior experience with. We even received the Synechocystis we work with from a research group in Stockholm. Also, we received input from various researchers when it came to finding the exact proteins for the different parts of our project. But in order to make our end product even better, we've been in contact with some of the potential users, more specifically farmers.
Yes, and from the farmers, we got validation that there is a demand for our product, even here in Sweden. We got a lot of ideas and feedback that helped us form an initial idea of our industrial setup. And it also affected both our wet lab experiments and our modeling work by shifting our attention to studying the efficiency and costs of our desalination process. Overall, the feedback was very positive, and some of the farmers were even interested in becoming future customers if we decide to commercialize our projects. However, since a synthetic biology solution for desalinating seawater has never been upscaled before, as far as we're aware at least, we decided to contact the industry for some inputs.
And based on both input from the industry as well as the feedback from the farmers, we made a draft of how we would like our upscaled setup to look like. Water would be pumped in from the sea into a water tank with an attached bioreactor containing our Synecocystis culture. The accurate amount of Synecocystis would be released into the water tank to allow desalination to take place. Then, the desalinated water and the bacteria would be released into the bottom tank, but the Synecocystis cells would get stuck in the filter separating the two tanks. From there, the desalinated water would be transported via pipes to the ultimate agricultural users, and the biomass would be used for biofuel production and inoculation.
So in conclusion, our project Sea Salt consists of a lot of different aspects and helping hands that come together to close the sea salt cycle. And we in the Linköping iGEM team are very excited to see our project evolve and reach its full potential so that we can hopefully offer a solution to a worldwide crisis. So we want to thank you for your attention, and we're now happy to answer any of your questions.
Thank you, team, and Gopin. That was a fantastic presentation.
Thank you.
The floor is open for comments or questions.
A great presentation, extremely slick and wonderful graphics. I'm practically ready to sign on board. So, really beautiful. And one of the first questions that come to my mind, I'm sure you must have thought about this a bit, was when you think about the amount of algal or cyanobacterial biomass that's in a liquid system, the amount of biomass relative to the volume of the liquid is quite small. And so have you thought about how much of a concentration of these ions you really need to get into your cyanobacteria in order to get a decent desalination?
Yeah, so that's one of the things that we're experimentally studying now in the lab, seeing how much biomass we would need for a certain amount of ions being removed from the surrounding water, to see if the industrial setup would look more like it does now, or if it would have to be more of a step-by-step solution where we take it in phases, basically. So we're studying that, and our modeling team is also working on creating a model for that, basically. I don't know if that answers your question. We don't have any definite answers yet.
Yeah, and by the same token, how much can an individual cell tolerate of these ions before you salt out proteins and the enzyme stop working?
Yeah, exactly. That's one of the most pressing questions of our projects, and also a reason as to why we need to figure out how much they can tolerate before they either die or start releasing it back out, so that we can have a time, a time point of when to move the water to the next phase before they start releasing it back out, if that makes sense.
So do it sequentially, you would have multiple reactors?
Yeah.
Interesting. Yeah. So, I mean, the sea salt cycle that you portrayed is just so appealing. I really admire it. Nice work. And you picked a problem that couldn't be more important. Potable water and water for agriculture is going to be a huge, it already is a huge problem. So I guess the other question is, in that cycle, when we're attempting not to waste anything, you're using salty cyanobacteria as fertilizer. Is that an issue for crops?
Well, we would, separate the cyanobacteria from the water before it's used for agriculture.
Oh, okay. I'm thinking about the algal biomass. What do you do with that algal biomass after it's?
Oh, yeah. So, I mean, this is also not completely a finished idea yet, but we're hoping that we could use it for biofuel production. So we're reaching out to companies right now, discussing this to see what possibilities there are.
Oh, I misunderstood. I was thinking that was going to be fertilizer, but no, biofuels.
Yeah.
Okay.
Yeah, biofuel. Yeah.
Okay, so I think that you guys have chosen a really fantastic real world problem. So I will go the other way. Instead of trying to desalinize the lake, how about just having enough so that somebody in Africa doesn't have to travel 5 miles to get potable water? So I'm more thinking about the modeling aspect. This is so exciting. That's why when you were presenting, which was very well presented by two Alexanders, I guess, that maybe the bang for the buck is not in trying to desalinize world's water, but rather desalinize at a scale where people can drink water. Have you thought about that concept, and what would that look like?
Yeah, and I think that was like our initial aim, was to be able to provide drinking water. But as of now, like, you know, for iGEM, we just have a limited amount of time. So we, for this year, will only be able to desalinate the water. I mean, there's still other microorganisms in the water, so it's not going to be drinkable. But that's definitely one of the things that we would love to do in the future if we keep working with this project. Because that would require more steps. But yeah, that would be one.
Well, I mean, you can ask your colleagues from India or other developing countries in Africa that, yeah, the microbes are there, but people usually just go in and drink water from the sources that they have. But the major problem always is that it's too salty, especially if you are close to an ocean and things like that. So at the level of individual or a small group of individuals who do not have access to potable water, your approach may already be almost revolutionary. So you may want to think about it.
Yeah, that's a great point, actually.
Okay, and on the science side, I don't know whether you are aware of the fact that people have done this kind of rhodopsin, putting it in photosynthetic organisms. And one additional bang for the buck is that when you put those rhodopsins in, it enhances photosynthesis because it acts as an antenna. So maybe that's another aspect that you want to point out. Either, I do not know whether helorhodopsin will provide the same kind of advantage as with bacteriorhodopsin that people have done already. But if you are putting in a pigment protein, that acts as an antenna also. So you will enhance photosynthesis in synecosystem. So what thinking about that?
Yeah, thank you for that notes. We'll definitely write that down.
That's very
Yeah, and if you have problem in finding the appropriate literature, just drop me an email. I'll be happy to send it to you.
Oh, thank you. Thank you so much.
I just wanted to bootstrap on a comment that Himadri already made, which, you know, I think it could be a really nice selling point to do a little bit of the calculations for, well, what's the power consumption for sort of a normal desalinization process versus, you know, like, let's say you put some solar panels or something to collect a equivalent amount of light that you would need to grow your algae, right? It might be the case, and my intuition is that probably the solar panel is going to be more effective. But if you're talking about this sort of distributed idea where you're making a local concentration or a local production of drinking water, the need for all of the additional technology and expense of the setup could actually make a biological desalinization process much more attractive.
Yeah, we're definitely hoping to be able to, you know, get the energy consumption down. And since they're photosynthetic for the cultivation of the bacteria, we wouldn't need any solar panels either. Like the sunlight would be enough. So yeah, but those calculations I think would fit perfectly in our presentations.
Are there any last quick comments or any questions from your side?
Nice job, and a nice back and forth with the presentation as well.
Oh, yeah.
Thank you. Thank you so much. And thank you for having us.
Great. So I would request all the judges to fill the judging form. We have 5 minutes. So are the judges done with filling the form? Great. I guess yeah, the judging form is complete. So with this, we come to the end of day 1 of our symposium, and we hope you took away some valuable inputs from this event.
A big thank you to all our judges for being kind enough to take out time from their busy schedules and help us improve our project design. So I'm going to say this in on behalf of all of us, talking in front of all of you was a very overwhelming experience for all of us. Most of our project ideas have been inspired by your work, and we have learned so much throughout your papers throughout the year. It was almost like you were guiding us at every step of our project. I know that all of us are trying really hard to act normal right now around all of you, but this is truly unreal for us, and we are really glad that all of you could make it tonight and join us as judges. And before concluding today's event, I'd like to ask our judges if they have any general comments for all our teams.
Just thank you so much for including me. It was really fun to hear all these creative project ideas, and I had a really enjoyable time. So thank you, everyone.
Yeah, I want to thank everyone for their presentations, and want to say that hearing the fabulous projects that you're engaged in gives me hope for our future. Gives me a lot of hope for our future.
Yes, thanks for inviting me. You know, usually my Saturday mornings are taken on doing all kinds of other things, right out there, mowing grass and things like that. This is so exciting, and maybe someday, you know, we will have the true benefits of the projects that you guys are starting, and I'm looking forward to that day. But most important, as has already been mentioned, is that this is the right type of project for the era that we are in, the era that we have given you guys, that you have inherited now, and you are going to bring in the solutions for that. So thanks.
Yeah, keep up the great work.
Absolutely. Thank you.
Thank you so much. You are our inspiration. Like, I know all of us want to, like, all of us in this meet want to say this to all of you. Thank you for fueling our dreams, and we try our best to follow your steps. And yes, I'd like to thank and congratulate all the collaborating teams. We finally had a successful day one. Before concluding, I'd also like to thank all the iGEM teams for joining. We hope to see all of you tomorrow for day 2, which will be packed with interesting talks, workshops, games, and more. See you. Stay safe.
Stay safe, guys.
Bye.
Bye-bye.
Bye-bye.
Thank you so much.
Thank you all. Bye.
Bye. Thank you for joining.
Thank you. Bye.
Goodbye.
Bye.
Bye.
Thank you. Bye,
Bye.
everyone.
Thank you.
Thank you.